Welcome to Science News Review for July 17, 2026. Today, we explore how gravity, or the lack of it, shapes our biology, how scientists are looking to stop genetic diseases at their source, and the discovery of a rocky exoplanet that might just be holding onto its atmosphere. We begin with the physiological challenges of long-duration spaceflight, specifically the muscle wasting that affects astronauts. While we have long known that weightlessness impacts how genes are expressed, new research suggests the problem goes much deeper, reaching into the actual construction of proteins within our cells. In a study published in Nature, researchers looked at what happens to mitochondria, the energy-producing powerhouses of the cell, when gravity is removed. By culturing human cells and a type of worm called Caenorhabditis elegans aboard the International Space Station, scientists observed a significant drop in mitochondrial protein synthesis. It isn't just that the instructions for these proteins, the messenger RNA, are being sent less frequently. It is that the actual assembly process, where ribosomes build the proteins, is being disrupted. This decline was observed within just 24 to 48 hours of exposure to microgravity, and continued to drop through the 72-hour mark. To confirm these findings on Earth, researchers used a device called a clinostat to mimic the effects of reduced gravity, and the results were consistent. Perhaps most interestingly, the study identified a specific molecular pathway for this effect. It appears that the mechanical action of cell adhesion, the way cells physically stick to one another, is what communicates the presence or absence of gravity to the mitochondria. When that physical tension changes in space, the cell's ability to build essential mitochondrial proteins falters. For future missions to Mars and beyond, understanding this mechanical link could be vital in developing ways to prevent the profound muscle loss that astronauts currently face. Moving from the microscopic mechanics of the cell to the fundamental sequences of our DNA, there is a new way of looking at the progression of Huntington's disease. As many know, Huntington's is a progressive condition caused by a mutation in the HTT gene, specifically a repetitive stretch of DNA known as CAG repeats. While inheriting the mutation determines if a person will develop the disease, the timing of when symptoms actually appear is driven by a process called somatic expansion. This is where the body's own DNA repair machinery accidentally causes those repetitive sequences to grow longer over time. Once these repeats reach a certain threshold, they trigger a toxic phase that leads to neuronal death. For years, much of the research has focused on trying to lower the levels of the toxic Huntington protein itself, but a new direction in genetic medicine is looking upstream, trying to stop the mutation from expanding in the first place. Researchers are investigating the mSci gene, which governs how DNA is repaired. In animal models, blocking this gene has shown the ability to slow or even stop the growth of those repetitive DNA sequences. By targeting the repair mechanism rather than just the protein produced by the error, scientists hope to halt the disease before the toxic phase even begins. However, this approach is not without its hurdles. Because mSci3 is part of the body's natural DNA repair system, suppressing it carries risks, such as the potential development of intestinal polyps. Currently, clinical strategies are divided between two main paths: lowering the Huntington protein or modulating DNA repair to stop the mutation at its source. While some gene therapies have shown promise in slowing the disease, they often require invasive brain surgery and do not stop the progression entirely. Experts suggest that the ideal treatment might eventually be a combination of both approaches, though such a dual-pronged therapy is likely still years away from clinical reality. Finally, we turn our attention to the search for habitable worlds, where astronomers have found an unexpected candidate in a rocky exoplanet known as LH-740b. Located in the habitable zone of a nearby M-dwarf star, this planet is about 1.7 times the size of Earth and 5.6 times as massive. For many planets orbiting M-dwarf stars, the intense radiation and frequent solar flares from their host stars tend to strip away any atmosphere they might have. But LHS-150b appears to be an exception. Using the Magellan Clay Telescope, researchers detected excess helium gas streaming away from the planet during a transit. This provides direct evidence that an atmosphere exists. While the planet is losing some helium, it is doing so at a much lower rate than larger gaseous mini-Neptune planets, likely because it is cooler. This lower rate of loss is significant because it suggests that LHS-140b is uniquely stable for its size, allowing it to hold onto its air where other similar worlds would be stripped bare. Calculations suggest that this planet has held onto its atmosphere for more than 3 billion years and could remain stable for another billion. This stability is crucial. It means the planet could be retaining heavier gases like nitrogen or carbon dioxide, or perhaps even harboring a global ocean of liquid water. LH740b has emerged as a rare, concrete example of a rocky planet in a habitable zone that might actually possess the conditions necessary to support life. The next step for astronomers is to use the James Webb Space Telescope to look even closer, attempting to characterize the exact molecular makeup of this exotic atmosphere and determine if it is truly a world capable of sustaining liquid water. Whether it is the microscopic dance of proteins in a cell or the vast helium-streaked atmosphere of a distant world, science is revealing that the stability of life and planets often depends on the smallest, most persistent details.